Hello everyone, my name is Claudio Piselli and today I will tell you who will be the protagonist of the next pandemic. During the last decades, the World Health Organization acknowledged the growing problem of antibiotic resistance and listed the most common infection species under the acronym of ESCAPE. Multi-drug resistant bacteria survive broad-spectrum antibiotics and require higher usage of specific ones. All gram-negative bacteria also have an intrinsic resistance against antibiotic treatments that occur with the highly selective filter represented by their membrane. During my PhD, I focused in cracking the chemical barrier of Pseudomonas aeruginosa, which basal antibiotic resistance relies in the composition of porin reconstituted in its outer membrane. In fact, it lacks of general diffusion channel and expresses a set of narrow and specific porins, among which there are OprO and OprP. They are highly selective for pyro and monophosphate respectively. These beta-barrel trimeric porins are highly homologous and their structure shows three different regions. I studied the lysine cluster present in the periplasmic cavity because the characterization of the ion path across any porin is the first step for understanding its performances, and so to design a new generation of molecules that can selectively go through them. It has been demonstrated that OprO and OprP are highly induced during infection when the bacterium is in contact with lung epithelial cells. So using them as a specific uptake way for phosphonates antibiotics can counteract the infection at the very beginning through a favorable pathway. Speaking of which, we analyzed the two phosphonic acid-containing antibiotics, fosfomycin and fosmidomycin. They are approved drugs very active against gram-positive and negative bacteria, and even protozoa. We studied the permeation of fosfomycin and fosmidomycin via the reconstitution of OprO and OprP into the polarized lipid bilayer. The ion current flowing through each channel is represented by a step. The addition into the aqueous solution of antibiotics resulted in decreasing the ion current going through the channel in a concentration-dependent manner as a result of the antibiotic binding to the channel's binding sites. This is the prerequisite for the translocation and can be confirmed in both single-channel and multi-channel analysis. In this study, we demonstrate that the natural selectivity of any other membrane porin can be used to improve the uptake of the most suitable antibiotic across the bacterial outer membrane, which is the base for the optimized medicine of the future.